Today we are discussing a very important topic in renal. Our topic today is called acid-base balance and its disturbances. This topic is related to biochemistry and it has many details, but in this video I want to focus on physiological points which are so important for medical students. You know that what is the difference between acid and a base? Whatever, either it is proton donor or proton acceptor. And all of us know that acid is symbolized by hydrogen plus, H plus, any acid. We have many different types of acids, either fixed acids or volatile acid. I mean by a volatile acid which can be expired by the lung, as carbonic acid will be dissociated into carbon dioxide plus water. What about fixed acids? Fixed acids are not dealt by the lung. The kidney has to face this acid. For example, sulfuric acid, phosphoric acid, acetoacetic acid. All of these are not the responsibility of the lung to deal with. So this is the function of the kidney to neutralize these acids. Today we want to focus on regulation of acid-base balance. What is the function of lung and what is the function of kidney? But before dealing with functions of both organs, first of all, I want you to stress on Henderson-Hasselbeck equation. Henderson-Hasselbeck was a Dutch scientist who puts an equation for the pH. pH is the symbol of potential of hydrogen ion, the abbreviation of potential of hydrogen ion, the power of hydrogen ion. pH is direct proportional to logarithm ratio of salt as a numerator and acid as a denominator. Any salt I mention it today in this video, I mean sodium bicarbonate. So the numerator will be HCO3. I will not mention NaHCO3. It is understood HCO3 as a numerator over an acid. You will tell me which acid you are talking about. I am talking about carbonic acid. Doctor, why? Because bicarbonate buffer is the most effective buffer in our body, and any effectiveness of any buffer depends on two data. Number one, the concentration of its acidic part and its alkaline part. Any buffer acts as a double-edged weapon. It has an acidic part which is called carbonic acid and the alkaline part which is sodium bicarbonate. What is the wisdom of the double-edged weapon? Suppose that we have high alkali in plasma, we want to neutralize it by binding to carbonic acid. I mean by neutralization, you change the strong alkali into weak one. The reverse, suppose we have high or strong acid concentration in plasma, you should bind it to, very important, to a weak base. I want to change it into a very weak acid. It's very important. So neutralization means change the strong into the weak, either strong base into weak base or strong acid into weak acid. So we have this ratio, log salt over acid. All of us know that logarithm will minimize the mathematical value. So what about the denominator? The denominator will not be carbonic acid. Doctor, why? You said denominator is an acid. Because this carbonic acid rapidly dissociates into carbon dioxide plus water. So the denominator will be CO2 pressure, because carbonic acid is direct proportional to CO2 pressure. If CO2 pressure increases, carbonic acid will increase. You know that CO2 is 24 times soluble in water more than oxygen. So our ratio, which is so important to talk today, will be numerator is HCO3, denominator is PCO2. Doctor, you said that pH is direct proportional to log salt over acid, but we want direct proportional to put it into equation. We want to make it simple. In math, they told us when we were young students that direct proportional equal constant. What is the constant today? Any buffer has got its constant. You have many buffers: phosphate, hemoglobin, and bicarbonate, for example. Today we are going to focus on bicarbonate buffer. Although phosphate is more plentiful inside the cell, but I don't say it is the most effective buffer. Doctor, why? Because the acidic part of the phosphate buffer or the alkaline part cannot be regulated by lung and kidney. You should have two factors. Both acidic and alkaline part of the buffer should be regulated, and the concentration. Not only one factor to make you dominant or have upper hand. It's so important. The pK is called the constant of the buffer. And I want you to imagine, although constant start by the letter C in English, it is written K, because in Deutsch language C is called K. Constant start by the letter K in Deutsch language. So pK, because who makes the equation is a Deutsch one, Henderson Hasselbeck equation. Today we want to focus on what is the function of lung to regulate this balance. You know that lung will deal only with volatile acid, carbonic acid, which can be expired. So if the lung wash out carbon dioxide in normal level, you have a normal lung. But suppose your lung doesn't wash CO2, what will happen? CO2 concentration in plasma will increase and it will be more soluble in water. You know that 90% of plasma consists of water forming carbonic acid. So acidosis occur. The term osis means increase. You have acid level higher in plasma. Suppose the reverse, your lung wash out carbon dioxide more than normal, above normal, you will suffer. CO2 level, PCO2 will decrease, carbonic acid will decrease, so alkalosis occur. There is a balance between acid and alkali. If your acid level is going to decrease, the alkali level will seem higher in concentration than the acid, causing alkalosis. So here you have a problem. Your question to me now, doctor, explain the mechanism of washing out carbon dioxide in normal. Any acid is symbolized by hydrogen ion. You know that hydrogen ion does not cross the blood-brain barrier. So it will stimulate certain receptors in the aorta called aortic bodies and carotid bodies. Carotid bodies is present in the common carotid artery. These receptors are far away from respiratory center in the brain, so they are called chemoreceptors, related to sensitivity to chemical change, and they are far away from respiratory center in medulla, so they are called peripheral chemoreceptor. This receptor will send signals across vagus and glossopharyngeal nerve to respiratory center in medulla to be activated, and this center will send signal to your respiratory muscles to be more active, causing hyperventilation. Wash out carbon dioxide and return its pressure in plasma back to normal. Every time in homeostasis, we focus on this statement, back to normal. This is the mechanism of action of lung to deal with the balance. What about kidney? Kidney can behave in three mechanisms, and the three mechanisms share one idea. What is it, doctor? Kidney wants to reabsorb sodium bicarbonate back to plasma. You know that the kidney will have sodium bicarb in its lumen across the filter. This filter, you want to return it back to plasma. So the question now, from where we will bring sodium bicarbonate? And I will give you another question. From where you will bring sodium, not HCO3. And you will know why now. Because sodium is the main determinant, and HCO3 will be passive to it afterwards. But before discussing the mechanism, I want you to imagine something. My hand is the cell of the kidney. I have right hand and left hand. Next to my right hand, here is the lumen, and next to my left hand, here is the plasma. So I have a box as if between my both hands. Imagine there is a box, and you have next here plasma, and next to inside to the right hand you have the lumen. What will happen? First of all, I want you to imagine from where HCO3 will come. Very easily. The two hands are the cell. Here I have got carbon dioxide in plasma. Carbon dioxide enter from plasma, which I point with my finger, to inside the cell by diffusion from high concentration in plasma to low concentration inside the cell. So here we have CO2. What will happen next? CO2 is soluble in water inside the cell by the effect of carbonic anhydrase enzyme, forming H2CO3. I will form carbonic acid inside the cell. Carbonic acid is going to dissociate into two elements like this. One is called hydrogen, H+, and the other is called bicarbonate. I want you to imagine something. H+ is nearer to what? To the lumen. HCO3 is nearer to the plasma. I wanted to get rid of hydrogen ion. Getting rid means what? Moving hydrogen from the cell to the lumen, which is called secretion, is very important. But hydrogen ion to transport from lumen cell to the lumen, it needs exchange with another positive ion. This positive ion in exchange will be sodium. So in the lumen, I will have sodium. Sodium coming from different sources. First source, sodium bicarbonate itself, which I wanted to move into plasma. It enters the lumen. Yes, kidney makes filtration of plasma. 3.5 liter is filtered 60 times every day, or 3 liters are filtered 60 times every day, as if the kidney is filtering 180 liter plasma free without paying any money. This is the God power. So NaHCO3 enter the lumen. It will dissociate into Na plus sodium, HCO3 negative. Na plus in the lumen will enter the cell, and in exchange with hydrogen, which will move in its place. This is called secretion. what happens? Hydrogen enters the lumen to bind to HCO3 to form carbonic acid, and the carbonic acid will dissociate into CO2 plus water inside the lumen. CO2 in the lumen will enter the cell again to complete a cycle. What do you mean by a cycle? CO2 enter again the cell to react with water, forming carbonic acid, dissociating into hydrogen and bicarb, and you will complete a cycle. It is called carbon dioxide cycle. This is very important because you want to make this cycle continuous, not only temporary. So sodium enters now the cell. When it enters the cell, it will move out by sodium potassium pump, moves out, and this sodium will attract negative ion afterwards called HCO3 in the cell. So sodium bicarb is formed in the plasma. The name of this mechanism is reabsorption of already filtered sodium bicarbonate. We didn't sensitize sodium bicarbonate. It is already formed and moves in filter. Then we move it from cell to plasma as if you have got money. Instead of putting it into your right jacket, you will move it and put it into your left part of the jacket. So you didn't benefit any value. You know that sodium bicarbonate or bicarb act as an alkali reserve. Doctor, can you explain how? Yes. Sodium bicarb as a strong alkali will bind to lactic acid. Bind to lactic acid to form sodium lactate, which is a weak base, and carbonic acid, which is a weak acid. So here we neutralize strong acid, which is lactic, into weak acid, which is carbonic. This is alkali reserve, and we explain it before in respiration. Please return back to chloride shift phenomena. Bicarb concentration equal 22 to 24 milli equivalent per liter. We want to increase the concentration of this alkaline reserve, so we will move to the second mechanism. Inside the lumen, you have a new filter called disodium hydrogen phosphate, Na2HPO4. I will repeat it again. Na2HPO4. Na2HPO4 will dissociate into Na alone, sodium, and NaHPO4. Before I continue explanation, Na2HPO4 is sodium alkaline phosphate. Sodium alkaline. Na will move from the lumen to the cell, and on the contrary, hydrogen will move from cell to the lumen. Again, secretion occurs. We make exchange, which is called Antiport Mechanism. Hydrogen moving into the lumen will react with NaHPO4, forming NaH2PO4, which is sodium acidic phosphate. And don't be upset, because this acidic phosphate will not decrease the pH below 4.5. There is a law in the kidney. Any secretion or any filtration should not affect the pH in the lumen to be below than 4.5. Otherwise, secretion will stop and you will die. So here, the benefit is sodium move into the cell, then it will move out to the plasma, then pulling bicarb afterwards, forming sodium bicarbonate. The effectiveness of sodium bicarb concentration will be higher than the first mechanism, which is reabsorption of already filtered sodium bicarb. But till now, we didn't reach to the highest benefit of sodium bicarb concentration. So moving into the third mechanism, which is so important, ammonia. What do you mean by ammonia? There is a difference between ammonia and ammonium. Ammonia, NH3. What will happen now? Before telling you, you should know the journey of ammonia. It is secreted in proximal tubule lumen. It moves to loop of Henle, then reabsorbed from loop of Henle to plasma, then secreted again in distal tubule. So now we are talking about allocation. Distal tubule, how the kidney in its final destination can regulate acid-base balance. First of all, you should know that in the lumen you will have sodium, but now the source will be a different one. I told you NaHCO3 already filtered. Second source of sodium, Na2HPO4. Third mechanism, sodium comes from a new compound, sodium chloride, which is present in plasma. Do you remember? Isotonic, 0.9%. So sodium chloride will dissociate into Na plus, positive ion, chloride Cl negative. Again, sodium move from the lumen to the cell in exchange with hydrogen ion. Hydrogen now will react with Cl, forming HCl. HCl is a very strong acid. It is a corrosive acid which will decrease pH in the lumen below 4.5. Oh God! Secretion will stop. Hydrogen ion level will increase in plasma. Acidosis will be severe. It will end by coma. The heart will stop. Mitochondrial enzymes will be affected by high acid concentration. So death will ultimately occur. But God is kind for us. The mercy of God, He creates NH3, ammonia. NH3 is present here in the cell of distal tubule. So NH3 will cross from the cell to the lumen because the membrane is permeable to it to save the life of human, to save human life, because NH3 will bind to HCl in the lumen instead of making corrosive effect on the lumen. It will buffer it. It will form a new compound called ammonium chloride, NH4Cl. This mechanism will increase the effectiveness of sodium bicarb concentration in plasma. It will reach 500 milli equivalent per liter. I want you to imagine what I said before. I told you that if bicarb concentration in normal 22 to 24, now it will be multiplied above 20 times, reaching 500 milli equivalent per liter. And this is a very effective and the most important mechanism of kidney to regulate acid-base balance. I imagine that one of students will ask me now an important question. Doctor, NH4 can leave the lumen to enter the cell? No. Why? Because the membrane of the cell is not permeable to NH4. And this is described in physiology textbook by this term. Please concentrate. Diffusion trapping mechanism. Who will diffuse? NH3, ammonia. Trap means what? After it binds to HCl, forming ammonium chloride, you are not allowed to leave your place again and return back to the cell. No, your place as a buffer in the lumen, as if you are a soldier in a battle. Yes, I am not exaggerating if I tell you this is a battle of your life. If the kidney stops hydrogen secretion, you will die. So you should struggle to be alive. Your buffer should not leave its place. Look to the wisdom of God. Any buffer doesn't leave its place. For example, hemoglobin present inside red blood cell cannot leave its place. Potassium acts as a buffer inside red blood cell, cannot leave its place. Sodium as a buffer in plasma cannot leave its place. And when I explained blood lectures before, I told you that red blood cells are not permeable to any cation. Any positive ion attracted to negative pole of the battery cannot be permeable to red cells. It's not allowed to enter. Potassium inside the red cell to buffer the hemoglobinic acid H.HB to form potassium hemoglobinate and release the acid. Sodium in plasma form sodium bicarb to buffer lactic acid. Your question to me now, doctor, why you say lactic acid? because it is one of end products of the muscle metabolism. You know that glucose, if in an aerobic condition, it will form lactic acid plus 2 ATP. So every time we should deal with lactic acid. So at the end of this lecture, I want you to imagine the important points. What is Henderson-Hasselbeck equation? What is the effect of lung to maintain acid-base balance? And what is the effect of kidney? Kidney can reabsorb sodium bicarb by 3 different mechanisms. 1, reabsorption of already filtered sodium bicarb. 2 is reabsorption of sodium bicarb through disodium hydrogen phosphate to form titratable acid, which is sodium dihydrogen phosphate. 3rd is the ammonia. Ammonia secreted in proximal tubule, reabsorbed from loop of Henle, secreted again in distal tubule. And what do you mean by diffusion trapping? Another lecture, I will focus on what do you mean by acid-base disturbance. Acidosis, alkalosis. We have 4 types of acid-base disturbance, and we will know together the name of each disturbance and how compensation will occur, how the body will correct its error. I hope all of you have a benefit from this video. You have Dr. Mohamed Fayez with you, lecturer physiology, Ain Shams University, Egypt. I hope all of you to have best wishes. Good luck from me, and if you are not subscribed in this channel, please subscribe and you will continue with me these lectures together.